Welcome to the Mobilize and Restore webinar series, and thanks to those of you that have continued on from the research talk. In this portion of the webinar, our speaker, Jennifer Maier, will give a tutorial explaining how to use the personalization approach for your own data. And using a Colab notebook and OpenSim, she will guide the audience through the step-by-step process of model scaling and model personalization. And again, we'll take more questions at the end of the tutorial, and again, use that Q&A panel. And feel free to enter your questions during the talk. With that, Jennifer, please go ahead and start the tutorial.
Yes, I will share my screen. All right, so this is our Colab notebook. This notebook takes you through the steps to personalize a musculoskeletal upper body model, like I just explained in the research talk. This notebook also entails the scaling of the model that I only briefly mentioned before. And then the second step is personalizing the shoulder rhythm constraints. Again, we are providing all our model and data on SimTK, and the entire code base that is also used in this Colab notebook can be found on GitHub. The tutorial has 4 main parts. It starts with setup and installation, which you can see is already running here because this takes a little moment to install everything, but I'll go through it in more detail in a bit. The second part is that we provide all the files and inputs that we need. The third part is the shoulder model scaling. And then the last part is personalizing the shoulder rhythm constraints. All the sections are collapsed when you open the notebook, but we encourage everyone to extend the single sections and run the cells individually. So first, for the setup and installation, again, I already started this because this takes a while to install everything. The first thing we need to do is to install Conda so that we can use it in the Colab notebook, which has already happened here, as you can see by the output. What's currently running is that we want to install the OpenSim Conda package so we can use OpenSim in this Python environment. So this is running right now. It takes a few minutes, and you can see the output similar to as if you were installing it on your machine. After this, we have some additional functions that we use to define, for example, body and joint coordinate systems, or the score method I will explain in a bit, and for the model scaling and personalization. And we are just copying these over from our GitHub repository to be able to use these methods in the tutorial. And then lastly, we need to import, just as in every Python script, all the packages and scripts that we are going to be using. Okay, and this is still running, but I can go on and explain some of the next steps already. All right, for the files and inputs, we need data. In this tutorial, we are providing example data that you can simply download by executing this cell. Of course, you can also use this Colab notebook with your own data. And you can upload your own data simply here on the Files tab on the side by dragging and dropping it from your machine. Let's just check. This is still installing. Okay. The files that you will need if you use your own data for the scaling and personalization are the unscaled model file. We need the 5 calibration poses from the static calibration measurements in which we use this L-frame to palpate and measure the scapula. All right. This crash of the session is a known thing that happens sometimes, but the installations still went through. So don't worry about that. It also says that here in the text. But we have to rerun the import. And we have to rerun this section as well. Okay, sorry. Now back to the files and inputs. If we download the data now, sorry, we will see that they are downloaded here into this folder structure on the left. And the files that you see are exactly the ones that I just started mentioning. So we see the model OSIM file. We see the 5 calibration poses for each shoulder. So here for the left shoulder, the 5 files, and the right shoulder, the 5 files. We need the scale settings file for scaling the model. So this is just an XML file that you could also just load in OpenSim to use the OpenSim scale tool. And then for that, we also need the neutral pose scaling file that has all the markers that I will show in a bit. In this scaling file, we simply combined the neutral pose for the left and right shoulders to one file. But of course, if you have a measurement with all the markers, you can also use that. Okay. Next, we need to provide some input values. If you're downloading the data we provide, you don't have to change anything here in the next bit. But if you're using your own data, you need to update those fields before you execute the cell. You need to give the path to where the data is, which is here on the left, simply mobilized tutorials data and shoulder model personalization data. You need to provide the name of the unscaled model. So in our case, model.osim. These next three names of output models, you can simply choose how you want them. So we need a name for the scaled model, scaled and personalized model, and scaled and personalized model with clavicle elevation, independent clavicle elevation. I already mentioned the scale settings file, XML file, and the scaling file with the neutral pose and all markers. We need to give the time range that we use for scaling, where we recommend you use a very short time range. In this case, we're using from 0.2 to 0.21 seconds. We need the file names of the 5 calibration files for the right shoulder and for the left shoulder. And so simply the names of the files. And then we also need to define what are our markers called in the TRC files. So for example, our xiphoid process here in our data set is named PX, but if you named it processor xiphoideus or something, then of course change this field to that and do that for all the markers that are placed on the upper body. And the markers that are shown here are the ones that you will need to run this tutorial and do the personalization. So this is a long list of marker names. And then finally, I already mentioned in one of the questions for the tutorial, our scapular palpation tool, this L frame in black that is displayed up here on the right, had these little pins so we could more accurately place it onto the palpated positions. And compared to all other markers that are directly placed on the skin, this means that those markers on the L frame have a certain distance to the skin. And if that is the case for your data as well, simply check this box, and then we will simply orthogonally move the markers onto the skin. For this, we need the names of the markers on the tool, and we need the distance from the markers to the skin. So basically the length of the pins in millimeters. For us, it was 57 millimeters. Okay, now that we have all the inputs, we can execute the cell, and it will just store all those names. And on we go to the model scaling. This model scaling entails the use of an OpenSim scale tool, and then some corrections. On the one hand, a correction of the humerus scale factors, a correction of the position of tracking markers on the upper arm and forearm. If you want to use your own model scaling, please just go ahead and do that outside of the tutorial and upload the scaled model. But if you'd like to reproduce what we've been doing in our study, execute the script. Okay, so again, this is just a reminder of all the markers that we are using for the OpenSim scale tool. But before we execute the OpenSim scale tool, we need to pre-compute the scaling for the humerus. You might be wondering why we need to do that. So if we only use the experimental marker data of the single neutral pose you can see here on the left, and simply use the marker on the acromion and on the elbows to scale the humerus long axis, this often leads to excessive humerus lengths, which will cause problems when we then try to do motions that are with a higher arm elevation. And so you can see here, if we only use the neutral pose, the factor would be 1.15 for this one certain subject, but a lot lower if we used other poses, or somewhere in the middle for other poses. So we pre-compute the scale factor for the long axis using the SCORE method, so the symmetric center of rotation estimation method, to estimate the center of rotation for the glenohumeral joint, so basically the center of the humeral head. This method uses the 5 calibration poses that we measured and where we know where the scapula is by palpation. It defines the body coordinate system of the scapula and of the humerus in all those 5 poses. And then it simply solves this equation in the linear least square sense in order to get the position V of the glenohumeral rotation center with respect to the humerus coordinate system and U with respect to the scapular coordinate system. And now if we use this estimated center of rotation, our scale factors are a lot more uniform throughout the 5 trials. So we're using this scale factor, this updated scale factor. Okay, this next bit of code does exactly what I explained right now. So it estimates the U and the V by calibrating using the 5 calibration poses, and then computes one average scale factor for the right arm and for the left arm. And we can see that the subject we are using the data of here, their arm is a little shorter, so we can see that the scale factor is around 0.9 for both the right and the left arm. Now that we have pre-computed this scale factor, we can use the OpenSim scale tool to scale the provided model with the settings in the XML file and with the neutral pose. And once that is done, the model is saved here to our provided file name model_scale.osim. Because some of the bones were scaled anisotropically, we should also update the joint coordinate frames in the model based on the ISB recommendations for joint coordinate systems. And by executing this next cell, the file model scale.osim is overwritten with the updated ISB coordinate systems. And now as a last step, we needed to do another correction. So the OpenSim scale tool has a marker placer, which moves all markers that are not on anatomical landmarks to the exact position that they were in the trial, in the recorded trial marker data. So in our case, those are the tracking markers on the upper arm and on the forearm on both sides. And we do this because we might place them a little differently for between subjects. Now, the issue that we have here is that the marker placer executes an inverse kinematics. So it poses the model exactly matching the marker data. But because of the shoulder rhythm in the model, it actually cannot place it exactly onto the markers, onto the experimental markers. So we can see here that the shoulder is a little too high in the model, which will lead to an elbow and to a hand that is a little higher than in the experimental marker data. However, since the tracking marker here shown here is moved exactly onto where it is in the recording, it will be off relative to, sorry, my runtime disconnected. Let's see if this still works. Off relative to the measured marker, sorry, off to the model markers here in pink, if we compare it to the measured markers. So we do a manual correction step that computes the coordinate system of the elbow and moves the marker into the correct position. All right. And this is simply done in this cell that I just executed. Now, if you'd like to, you can download the scaled model by executing this cell and then clicking this download button, which will simply download it to your desk. Okay, now we have scaled the model. The next step is the shoulder rhythm personalization. In this process, as I just explained in the research talk, we are using the 5 calibration measurements to compute and update the bilinear constraint parameters for the shoulder constraints. To do this, we first need to load the model. We set the model name that we defined above, and we tell it what are the names of the constraints. These are simply how they are in the model. If you change those names, of course, change them here. The next thing we're going to do is, as I explained before, we use the 5 calibration poses. So the neutral pose, 180-degree abduction and flexion, and maximum horizontal abduction and adduction at 90-degree flexion. And then we use the palpated measurements and all the skin-mounted markers to compute the joint angles of the sternoclavicular joint, of the acromioclavicular joint, and the glenohumeral joint. And this is done in the background right here. We are again using these two parameters of the score method, so the glenohumeral glenohumeral rotation center. And once this is executed, we can see the joint angles plotted out here for the protraction of the clavicle, elevation of the clavicle, scapular tilt, protraction and upward rotation, the glenohumeral elevation plane angle and elevation. And for the 5 rows correspond to the 5 poses. So if we look, for example, at row 2 and 3, where we have a very high glenohumeral elevation, we can see that these angles with 160 degree almost are very high. Or if we look at those 2 last rows for the horizontal abduction and adduction, we can see that the elevation plane is negative for the abduction and very high and positive for the adduction. And then finally, we use these angles that we just computed to fit bilinear planes for each of the constraints to get our slopes S1 and S2 and the constant C. And so here this is a for loop for every of the 5 constraints. We are getting this coordinate coupler constraint from the model and getting the linear function out of there. This bilinear function fits a bilinear function to the coordinates 5 and 6, which are the glenohumeral angles, and then the coordinate I, that is the coordinate that we're looking at right now. So sternoclavicular or acromioclavicular joint. And then we simply update the coefficients with the fitted bilinear plane. And here you can see printed out the parameters S1, S2, and C for the 5 constraints in the model. So this is now done for the right shoulder. And this next cell that I already clicked to execute performs the same computations for the left shoulder. So we start by computing the constraint angles that will be printed out once this is done. And then we fit the bilinear plane to for each of the constraints to update the coefficients in the model. Okay, now this is also done. And then finally, we save the model by printing it to a file. And if we update here on the side, we will now see that we have this model scaled _ personalized, which is the personalized constraint model. And again, executing this cell to download the personalized model and clicking on this download button will download the model to your computer. And then the final and last thing in this tutorial is to create a model in which the clavicle elevation is unconstrained, so independent. In order to do this, we remove the constraint and then set the range of the clavicle elevation to reasonable values. And then once this is done, we can again download the model. We can see that it's stored up here and we can download it to our machine. Let's have a look at all the models we just generated. For this, I am going to show you OpenSim. So this is just the OpenSim visualizer. And this wide model you can see here is the plain default model that we started with. Now, if I also show the scaled model now in red overlaid with this, you can see we saw that the scale factor for the humerus was 0.9, so the arm is a little shorter. We can see that this subject had a little wider shoulders than the default model. So this is simply the scaling, but the constraints in the model are still the same. Now, let's look at the personalized model. So in the default pose, now the model shown in blue and overlaid, we can see that the personalized model has their default pose with the shoulders a little lower, which is also what we saw consistently over most of our subjects. And now if we move the arm of the scaled model, so we lift the arm up, for example, here the right arm, let's lift it to 90 degrees, and then also move the arm maybe around the body minus 70 degrees to the back. And now if we do the same thing for the personalized model, so again, lift it to 90 degrees and minus, what did I put? Sorry, let me check, 70, minus 70 degrees glenohumeral elevation plane angle. We can see the difference between the two models, the personalized and the generic model. And our the final comparison we can do is to show the model with independent clavicle elevation. And if we put this back into its default pose, we can see that the shoulders are higher again, but this is not because that's somehow implemented. This is simply because this model has now this additional coordinate of the sternoclavicular elevation, so it can move the clavicle up and down independent of the elevation angle. So yeah, now if we, for example, lift the arm up, we will only see a protraction of the clavicle, but we will not see any change in the height of the clavicles on the elevation. But all the other constraints are the same as in the blue model. Yeah, so this is the end of the tutorial.
All right, great. Thanks, Jennifer, again for the tutorial. We'll switch back to questions and go ahead again. If you have any questions, enter them into the Q&A panel. If there's ones that come in more related to the tutorial, I'll go through those, but it looks like we could probably go back to some of the ones that came in earlier. The first question is from Victor Ing To Hing. The personalized fits show great improvement, but there are some small inconsistencies with the measured bone orientations. Can you speculate what might be causing this or how the model can be improved?
That's a good question. Why this happens is because we are still only using 5 poses and we're doing a bilinear fit. So the 5 poses might not be enough to exactly represent all the dependencies. But again, as I mentioned before, we had this trade-off of how many poses do we want to record versus how much time, so the time needed versus how accurate can we be. And then the second thing is that we fit a bilinear plane, which is a good estimate based on the study that the generic model was based on, that a linear fit fits those dependencies well. But there have been other studies that try to fit different types of planes. For example, a quadratic dependency in one coordinate, for example. So yeah, just a linear constraint might not be enough to represent the entire complexity of the shoulder joint. But again, we are trying to go for simple and applicable while still giving good results versus too much effort to get a working model.
Yeah, sure. Next question is from Genwa El Masri. Nice study. In the first result comparison, the ground truth that you were comparing your model to was from the experiment, correct? And also, how many subjects were involved in your experiment?
Yes, so it's correct. The white model that I showed, for this model, we basically removed all the constraints and picked the angles of the shoulder joint in a way that the palpated measurement was matched as best as possible. So we actually did just compute inverse kinematics using this marker information. So this is, yeah, the best representation of our actual palpated positions. In our study, we had 10 participants. They were all young, healthy, and active. None of them were designated overhead athletes. Yeah.
And just, I'll jump down. There's a question later from Ji Chen about how you created that ground truth representation and whether you use Visual 3D for it. But just clarifying that it's this was all processed in OpenSim. So
Yeah.
The next question, actually, I'll jump down because one came in about the tutorial, and it's from Martin Warner. When you use the score method, do you optimize the marker data and constrain the distance between markers, i.e., do you use the OCST prior to determining the glenoidal humeral joint center?
I am not entirely sure what the
Yeah.
OCSD prior is. So I'm happy to talk about this following up. But so this is not the original implementation of the score method by Eric and colleagues, but this was from a paper that specifically looked at the shoulder joint, where you need at least 2 poses. We had 5, so we used all the 5 that we had to calibrate the method. for the computation then during motion, and then, sorry, not during motion, but in different poses. And then of course, in different poses, we did not have the scapular orientation. We only used that for the later comparison. So we only used the variable U for the humerus to compute the glenohumeral joint rotation center for those. But yeah, happy if you'd like to, you can follow up and we can talk about this later.
Yeah, for sure. Sorry about that, Vern. The next question is from Hansen Zhao. And maybe just like kind of reframe this question, but they're asking if using data that includes scapular markers on subjects, would you advise using a model such as Seth's thoracic scapular model, or personalization, personalizing a generic model using your process for analysis? So maybe speak to like maybe the strengths of using one versus the other for depending what your research question is. Yeah.
Yeah, so I would say yes, it depends on your research question. So I have also worked and looked at the model by AJ Seth. For everyone who doesn't know it, it implements the scapulothoracic sliding. It represents the thoracic surface as an ellipsoid and has one point on the scapula slide on the surface. And by this, it allows for very flexible representation of the scapular position. It also enables the shrugging, for example, of the shoulders. We decided for our purpose to go with the constraint model, but personalize it in order to not need any measurements during the motion to get a realistic, reasonable, I don't know what's the right word, representation of the scapular position. The model with the sliding works well. It also works well with just giving it an estimate of where the scapula is. If you include muscles in the model, they will hold the scapula in place. But yeah, it is very hard to obtain estimates or reliable estimates of scapular position during motion because, yeah, let's not even talk about measuring during the volleyball hit or the baseball pitch. Yeah, so it was kind of we could do either, but we decided to go for this one for the practicality of it and for our application.
Great. Yeah. And actually, the next question was talking about, have you considered bringing together basically the independent clavicle model with the ellipsoid constraints of the scapulothoracic joint and the potential challenges?
Yeah, I was thinking about that. I was thinking about things like compute the kinematics using the constraint model, then removing the constraint, going to the ellipsoid model for all dynamic analyses. This is something that I can still imagine doing, especially moving forward when we're going to sports analyses. It might lead to some issues with the OpenSim model because already the constraints in the shoulder make the optimization a lot harder to solve because so many constraints have to be fulfilled in the computations. It would be interesting to see what a combination of both could do, if it would like completely break, if it cannot fulfill both the shoulder rhythm constraints and the sliding on the ellipsoid. But yeah, definitely would be an interesting thing to look at.
Yeah, sure. And sorry, that question was from Marcel Moreau Rossi. Forgot to mention that. So let's see. Next question. From Heron Medeiros. Great presentation. Do you think the model for overhead athletes would differ between sides? So given the asymmetric nature of many overhead athletes.
Yeah, I think so, yes. And we did perform a study with volleyball hitters where we did the exact personalization scaling that I just explained in the tutorial. And you can definitely see that in most of the overhead athletes, the right shoulder is a little, sorry, not the right shoulder, their hitting shoulder is a little lower in their just default standing. We haven't measured for them like internal external rotation that is known to be different for the hitting shoulder versus the non-hitting shoulder. But yeah, I'm sure in our analyses we will see very high external rotations in our athletes. So yeah, I'm excited to work on this.
I think we can squeeze one more in. Junan Lee. Nice presentation. How will you transfer the kinematic constraints to force or torque distributions in a dynamic simulation?
We can, yeah, so we can right now what we're doing is we added joint actuators to our model and to actuate the joints. It might be interesting what happens when we use our kinematics with constraints and then remove the constraints in order to analyze the motion with actuators, because otherwise the constraints will, because they're like infinitely strong, can take over some of the work. Yeah, but this is current work, and yeah, excited to see what comes out of there.
Right. All right. Well, unfortunately, we are out of time, so sorry for the questions we didn't make it to, but thank you again, Jennifer, for a great talk and for sharing this tutorial and your whole method. And thanks to everyone for all the great questions. Please be sure to click on the link that will appear at the conclusion of the webinar and take our survey. That'll help us improve the webinar series and choose upcoming topics. And this webinar series is supported by 2 grants from the NIH, including the grants to support our Mobilize Center and Restore Center. We're grateful for the support, which allows us to run these webinars and other events. And thank you to everyone for participating. We hope you'll continue to stay involved with our centers, and we'll see you again in a future webinar. Have a great day.